Thanks to the meeting organizers for the opportunity to share our research work here in the JGP EC Coupling Symposium. EC coupling starts at the neuromuscular junction, the site of crosstalk between motor neuron and the muscle. Because of the high energy demanding, both sides are packed with mitochondria. About 50 years ago, my lab started to study the skeletal muscle in ALS, a motor neuron disease. Our first discovery is that defective mitochondria with depolarized membrane potential first appear at the site of neuromuscular junction. ALS patients have severe muscle wasting. As no effective treatment, majority of ALS patients only survive 3 to 5 years after disease onset. Failure of respiratory muscle is a common cause of mortality in ALS. Studies from us and other groups support that muscle degeneration actively contributes to ALS pathogenesis. Oxidative stress is the marker of ALS skeletal muscle. Muscle fiber of ALS mice show enhanced ROS bright intensity. The increased mitochondrial red signal in the ALS muscle fiber indicates that oxidative stress likely due to excessive mitochondrial ROS production. However, the underlying mechanism is not clear. Additionally, both indicators do not provide quantitative evaluation of the ROS production and can tell whether ROS production varies during ALS disease progression. Pieces group established the CPYFP mitoflash as a dynamic ROS and MPTP indicator of mitochondria. Thanks to PEACE, we participate a small part of this work and had an opportunity to make a double transgenic mouse model with overexpression of CPYFP and an ALS mutation, SOD1 G93A. These double transgenic mice enabled us to follow the dynamic changes of mitoflash activity in live skeletal muscle fiber at the different ALS stages. The total flash area of fiber is increased in the 2-month-old mice. Remember this before ALS onset. Interestingly, after ALS onset, the muscle fiber showed a drastically increase in the duration of the mitoflash signal, but with a reduced flash fiber area. So the different property of mitoflash signal may indicate different MPTP status during ALS disease progression. As motor neuron withdrawal is a key pathological component of ALS, so we tested how mitoflash activity responded to derivation. As demonstrated here, muscle fibers responded to the cyanic nerve transaction with drastically enhanced mitoflash activity, which can be reversed by mitochondria by inhibition of MPTP opening with cyclosporin A. We question why mitochondria responded to derivation with enhanced MPTP opening. What we know is that derivation stops neuromuscular junction activity. That consequently eliminates EC coupling and the cytosolic calcium transients. We know mitochondrial calcium transit follows the cytosolic calcium transit. Absence of cytosolic calcium transit consequently eliminates the calcium influx into mitochondria. It is possible that loss of calcium influx into mitochondria is an initial trigger for excessive mitochondrial MPTP opening and ROS production. How about we add the cytosolic calcium transit back into the derivated muscle fibers? As you can see here, a brief electric field stimulation reduced mitoflash activity within seconds and minutes. Application of RU360 blocked the mitochondrial calcium uptake. Consequently, the field stimulation no longer had a significant effect on the mitoflash activity. This is further confirmed when used mitosox red to measure mitochondrial ROS level. So ALS and muscle disuse eliminate neuromuscular junction activity and the EC coupling. That stopped the physiological calcium influx into mitochondria, leading to increased MPTP opening and ROS production. It is known that long-term muscle disuse leads to elevated resting cytosolic calcium level, which cause mitochondrial calcium overload and excessive ROS production. Study from Zizi's group suggests that de novo expression of calexin gap junction channels may be responsible for the calcium influx during long-term derivation. We speculated that oxidative stress may damage sarcolemma integrity, resulting more calcium leakage and excessive mitochondrial ROS production. That could form a vicious cycle, needing muscle wasting under the condition of absence of AC coupling. As diaphragm muscle is so important, we went on checking the membrane integrity of the diaphragm muscle of the ALS mice. Evans blue dye was injected into the mice. 30 minutes later, the diaphragm muscle was collected from the mice with or without a modest rounding and examined on the confocal microscope. The diaphragm muscle of ALS mice show membrane leakage at the age of 2 months old before ALS onset. The 30 minutes rounding exacerbate the diaphragm membrane damage dramatically. The sarcolemma damage is also observed in the 4-month-old mice after ALS onset, and other muscle types like TA, for example, TA is also involved. As mentioned earlier, the mitochondrial defect appeared first at the neuromuscular junction of ALS muscle fibers. Naturally, we would ask if the neuromuscular junction is an active site of membrane injury. We applied both BTX and FM143 as a cell membrane impermeable dye to a FDB muscle fiber. FM143 dye accumulated at the neuromuscular junction. In the Y-type fiber, FM143 dye stayed at the neuromuscular junction. No intracellular signal were detected in the Y-type fiber. In the ALS fiber, the dye entered the fiber through the neuromuscular junction. As you can see, the dye diffused intracellularly away from the site of the neuromuscular junction. How could this happen? A series of studies from J&J Mouse Group demonstrated MG53 as an essential component for muscle membrane injury repair. As illustrated in the review article by Cooper and McNeil, MG53 is identified molecule so far that first arrives at the injury site within 2 seconds. Thus, we examine whether MG53 is involved in the loss of neuromuscular junction integrity. We made 2-month-old wild-type and ALS mice to run for 30 minutes, and then immediately took out the FDB muscle fibers for IHC. MG53 formed well-defined patches at the neuromuscular junction in the wild-type fiber. In the ALS fiber, MG53 also translocated to the neuromuscular junction. But the patch at the neuromuscular junction could be fuzzy, and some intracellular aggregates start to appear. At the late stage of ALS, there are MG53 aggregates at the neuromuscular junction, which is in sharp contrast to the wild-type fiber. We then compared the expression pattern of MG53 in skeletal muscle of ALS and wild-type mice. They are homogeneous pattern of MG53 in wild-type diaphragm muscle, while the G93A diaphragm showed aggregation and the patterns of MG53 after the 30 minutes of running. See. At the late stage, all examined muscle types showed MG53 aggregation in ALS mice, identified in diaphragm, EDL, solus, and TA. With the support of the target ALS Hume postmortem tissue core, we examined diaphragm and posterior muscle samples from both sporadic and familial ALS descendants. Here are non-ALS Hume muscle controls. Only review a few MG53 aggregates. Remarkably, MG53 aggregates were identified in all tested Hume muscle samples. Familiar ALS, sporadic ALS, different ages, diaphragm, different ages, Hume posterior muscles. It looks like that the endogenous MG53 lost its membrane repair function. Thus, we tested if application of exogenous recombinant MG53 protein could preserve that muscle membrane integrity in ALS muscle. We first performed in vitro tests to evaluate the FDB fiber membrane repair function following a laser-induced damage. Compared to the wild-type fiber, G93A fibers showed higher laser-induced membrane injury. See, G93A, wild-type. In the presence of MG53, the G93A fiber showed a reduced intracellular accumulation of AFM143 dye. Here, G93A with MG53. While application of the same amount of BSA was ineffective. The BSA here. Remarkably, the application of MG53 in the medium for 12 hours significantly reduced the mitochondrial ROS production in G93A fibers. G93A with MG53, G93 with BSA. We next IV injected MG53 Alex or BSA Alex as a control to the G93A mice immediately before the running. MG53 Alex, BSA Alex. After 30 minutes of running, the diaphragm muscle were immediately collected for live cell imaging. Mice treated with MG53 Alex showed prominent membrane patches of MG53. That's the membrane patches, indicating MG53 can target to the injured site of the diaphragm membrane. In contrast, the diaphragm of MG53 mice received BSA Alex showed a diffused pattern, indicating that BSA could not form membrane patches. The portion of fiber positive for the intracellular accumulation of EB was significantly reduced in the diaphragm muscle of MG53 treated mice. Our goal is to develop MG53 as a treatment for ALS. Thus, we evaluated the efficacy of MG53 in G93A mice after ALS onset at the age of 3 months. We can quantify the neuromuscular junction innervation in a whole diaphragm muscle using BTX and antibodies against neurofilament for axon terminal and skeletal facing for the release vesicles of axon terminal. In Y-type diaphragm, axon terminals make contact with all the neuromuscular junction, and the post and the presynaptic part, BTX, CYP, overlap perfectly. In the 3-month-old G93 diaphragm, neurofilament start to disappear, and completely or partially derivated neuromuscular junction were identified. See, missing CYP signal, partially CYP signal. Importantly, 2 weeks of MG53 IV injection slowed down the derivation process in the G93A diaphragm, see, innervated area and well-innervated neural junction in the G93A diaphragm received MG53 treatment. There are also significant more numbers of the survived motor neuron in the spinal cord of MG53 treated mice. See the motor neurons identified here and the motor neurons identified here. And the motor neuron number is slightly improved. In our pilot study, the ALS mice received 4 weeks of IV injection of MG53. Those mice survived longer. So those mice survived longer. And the body weight returned during the treatment. The benefit role of MG53 also holds for both genders. You see, the chi-square test. So they are a calcium-mediated coupling between neuromuscular junction, mitochondria, and the sarcolemma integrity that is essential for normal muscle function. Defects in this coupling could form a vicious cycle that promotes muscle wasting in ALS. Our future research goal will aim to break this vicious cycle by targeting mitochondrial function and preserve muscle membrane integrity to alleviating neuromuscular degeneration in ALS. I would like to thank my current and former lab members for their contribution on this study. And to Dr. Jian Ma from the Ohio State University, we collaborated on MG53 study. To Dr. Peace Chen from Peking University, we collaborated on MitoFlash study. To Dr. Lyle Oshaw from Johns Hopkins University, we collaborated on the human ALS study. And to Dr. Eduardo Rios from Rush University, guided me to EC coupling research field. Thank you.